Welcome to Nottingham British School, the home of science, maths and technology. I'm Dr. Zia Zamir. In this lesson, we're going to discover what enzymes are, explore how they work, and learn why they're needed for your cell's day-to-day functions. So let's start with definitions of enzymes. Enzymes are proteins that act as catalysts with living cells. Catalysts increase the rate at which chemical reactions occur without being consumed or permanently altered themselves. A chemical reaction is a process that converts one or more substances known as reagents, reactants or substrates to another type of substance, the product, which is called the product. As a catalyst, an enzyme can facilitate the same chemical reaction over and over again. So let's have a look at the structure and function. Like all proteins, enzymes are composed of one or more long chains of interconnected amino acids. Each enzyme possesses a unique sequence of amino acids that causes it to fold into a characteristic shape. An enzyme's amino acid sequence is determined by a specific gene in the cell's nucleus. This ensures that each copy of the enzyme is as small as all others. On the surface of each enzyme is a special cleft called the active site, which provides a place where reagents can meet and interact. Much like a lock and its key, an enzyme active site will only accommodate certain reagents, and only one type of chemical reaction can be catalyzed by a given enzyme. Okay, for example, during the manufacture of hemoglobin, which is the oxygen carrying pigment to your red blood cells, a single atom of iron must be inserted into the center of the molecule to make it functional. An enzyme called ferroglitase brings the reagents iron and the empty molecule together, catalyzes their union, and releases an iron-containing molecule. This is the only reaction catalyzed by ferro. Keep in mind that enzymes can combine reagents, as in the synthesis of hemoglobin. They can split a single reagent into multiple products, or they can simply transform a single reagent into a single product that looks different from the original reagent. When reagents enter an enzyme active site, the enzyme undergoes a temporary change in shape that encourages the interaction between the reagents. Upon completion of the chemical reaction, a specific product is released from the active site. The enzyme resumes its original conformation and the reaction can begin again with new reagents. Many enzymes are incorporated into metabolic pathways. A metabolic pathways is a series of chemical reactions that transform one or more reagents into an end product. That is needed by the cell. The enzyme in a metabolic pathway, much like people passing over a pail of water along a bucket brigade, move a reagent along until the end product is produced. A metabolic pathway can be quite short, or it can have many steps and multiple enzymes. The metabolic pathway that converts an amino acid found in a dietary protein called tryptophan to serotonin, a chemical that is necessary for normal brain function, is only 2 steps long. In order to function, many enzymes require the help of cofactors or coenzymes. Cofactors are often metal ions such as zinc and copper, iron or magnesium. Magnesium is one of the most common cofactor, activates hundreds of enzymes, including those that manufacture DNA and may help metabolize carbohydrates. Many coenzymes are derived from vitamins. In fact, one of the main reasons you need vitamins in your diet is to supplement the raw material for essential coenzymes. For example, vitamin C is needed by the enzyme that produces collagen and builds healthy skin. A coenzyme derived from vitamin B12 is necessary for synthesizing the insulation around your nerve cells. And vitamin B6 based coenzyme is vital for producing serotonin. Coenzymes and cofactors bind into active sites of enzymes and participate in catalysis. but they are not generally considered reagents, nor do they become part of products of the reaction. In many cases, cofactors and coenzymes function as intermediate carriers of electrons, specific atoms or functional groups, but they are transferred during the overall reaction. So okay, why do we need them? Why do we need enzymes? One of the immutable laws of nature is that energy is required to initiate a chemical reaction. After all, if you want to change something, you have to put some work into it. For every chemical reaction, a specific amount of energy, the energy of activation, must be applied to the reagents before reaction will proceed. The energy of activation is like a hill between the reagents, and the product and the reagents must be pushed over this hill before a reaction can continue. For some reactions, the energy of activation is so high that your cells either cannot supply it or would be harmed by the release of heat as a reaction proceeded. In other cases, a given reaction might proceed at a pace that is normally too slow to meet the cell's metabolic demands. That's where enzymes come in. They lower the energy hill so chemical reactions can get started more easily and proceed in conditions that are cell-friendly. Enzymes, along with metabolic pathway, can also divide one high-energy chemical reaction into many steps, each requiring only a fraction of the energy needed for the whole process. Finally, enzymes can accelerate the rate of reaction millions times over, which is critical to cells that often need large quantities of particular product within just a few seconds time. So, if you look at the general control of this, the genetic control, let's say, we are looking at a complex process that is guided from within the cell's nucleus when more specific enzymes products are needed. Regulatory proteins bind into the cell's DNA, switch on, and the genes produce the enzyme. Conversely, when a particular enzyme or metabolic pathway begins churning out too much product, different regulatory proteins stop enzyme production by attaching to the cell's DNA and turning those same genes off. So, where are we today? Scientists are constantly discovering new enzymes. New discovered enzymes are named according to regions as they act on, followed by the actions they perform. The suffix ASE is added to the name to identify its enzyme. Anytime you see ASE at the end of a long chemical name, you can bet you are dealing with an enzyme. Thus, sucrose, which finishes in ASE, is a digestive enzyme that breaks down sucrose, which is table sugar, and alcohol is an enzyme that detoxifies alcohol by removing hydrogen atoms from it. In addition, enzymes are included in a subclass whose name describes the general type of reaction catalyzed by all enzymes in that subclass. For example, enzymes that transfer groups of atoms from one molecule to another would be classified as transferase. The enzymes that break the chemical bonds into a molecule by adding hydrogen would be called hydrolase. Every living cell contains hundreds or even thousands of enzymes, all interacting in a coordinated dance that keeps the cell function efficiently and, in the case of multicellular organisms like humans, working together for the good of the whole.